membrane structure results in selective permeability. Remember that plasma membrane is made up of phospholipid bilayer, and the phospholipid bilayer, the head of phospholipid bilayer is made up of choline and phosphate group. It is hydrophilic, and the tail of the phospholipid is made up of fatty acid, either saturated fatty acid or unsaturated fatty acid. Therefore, in selectively permeability, only hydrophobic molecule can pass through plasma membrane rapidly without using any transport protein. However, hydrophilic molecules, including ion, polar molecules, and large molecules, need proteins to transport them across plasma membrane. There are two types of transport protein. One is channel protein. The other one is carrier protein. This hydrophilic channel provided by channel protein will allow hydrophilic substance to pass through plasma membrane. One of the example of channel protein is aquaporin, and it is used to transport water. Channel protein do not change shape. Compared to carrier protein can change shape. It provides binding site for specific substance to bind to the carrier protein, and then it will be transported to the other part of plasma membrane. There are two types of transport across plasma membrane: passive transport and active transport. Passive transport involves diffusion, either simple diffusion that allow the substance to pass through plasma membrane without using any transport protein, or facilitated diffusion. This type of transport need help from transport protein, either channel protein or carrier protein. However, passive transport do not need any ATP. In active transport, ATP is needed to transport substance across plasma membrane. Generally, molecules have tendency to spread evenly in available space. Each molecule moves in random. However, population of the molecules move in a specific direction. At dynamic equilibrium, net of molecule movement across membrane is 0. This is the example of diffusion of one solute. We can see here that this is membrane that allows certain molecule to pass through it. And we can see that at site A, molecule of dye is very high compared to site B. So, since molecule move at random, however, population of molecule will move in specific direction. It move from high concentration site to low concentration site. So at this point, molecule of dye will move from side A to side B. And we can see that here, the molecule of dye is moving to side B. And at equilibrium, you can see that movement of the dye to side B or side A is similar. So net movement of a molecule from A and B side is 0. This is example of diffusion of two solute, and we can see that at side A, yellow dye is high compared to side B. There is no yellow dye. And on the other hand, purple dye is very high at side B, and there is no purple dye inside A. Therefore, both dye molecule will move at side A and side B to reach equilibrium. And you can see that the yellow dye will move from A side to B side, and purple dye will move from B side to A side until equilibrium is reached. So the simple rules of simple diffusion is substance will diffuse from high concentrated area to less concentrated area. And we say that it diffuse down concentration gradient from high concentration to low concentration. Or sometimes we say that it follows concentration gradient. And no energy is needed, no ATP is needed, and it is spontaneous process. One of the example of simple diffusion is osmosis. So osmosis is diffusion of water molecule across selectively permeable membrane. So since it is diffusion, it diffuses from high concentration of water to low concentration of water. On the other hand, you can say that it diffuse from low solute concentration to higher solute concentration until equilibrium is reached. So let's see here. This is U tube, U-shaped glass tube, right? Atau kita panggil U tube. Okay, tube U. And you can see that this is A side, this is B side, and this is the membrane. Okay, and this membrane has pore. So you can see here, this pore only allows water molecule to pass through, not the glucose molecule. So only water can pass through, no glucose can pass through. So this is the solute. Glucose is the solute. Alright, let's see here. At A side, sugar molecule is low. At A side, sugar molecule is low. At B side, sugar molecule is higher. Remember, in osmosis, water move from low concentration of solute to high concentration of solute. So in this case, water move from A side to B side. So you can see after certain time, the water level at B side is higher compared to water level at A side. Osmosis will affect tonicity. Tonicity is the ability of solution to cause a cell to gain or lose water. In isotonic solution, solute concentration is the same as inside of the cell. Hypertonic solution have greater solute concentration than inside of the cell. So the cell will lose water. On the other hand, hypotonic solution have solute concentration that is less than inside of the cell. Therefore, the cell will gain water. Osmosis can have different effects, Osmosis can give different effect towards the different cells. So example here is animal cell. When placed in hypotonic solution, the cell will gain water. Water will enter the cell. Alright, the cell will swell and eventually lyse because animal cells do not have cell wall. So when it receives a lot of water, eventually it will lyse. If the cell is put in isotonic environment, net movement of water is zero, or we say that no net movement of water. So water flow inside the cell and outside of the cell at similar rate. So the volume of animal cell is stable. It has normal shape. On the other hand, if the cell is put in hypertonic solution, it will lose water towards the environment. Water will move out. So the cell will become shriveled and it will probably die. In plant cell, on the other hand, if the plant cell is put in hypotonic solution, it will receive water. However, since plant cells have cell wall, it do not lyse. It will become turgid. And this situation is normal for plant cell. If the plant cell is put in isotonic solution, similarly to animal cell, no net water movement, no change in cells volume, and the cells become flaccid. The plant will wilt. In hypertonic solution, the net outflow is higher, so water will move out. The vacuole shrinks. Plasma membrane is pulled away from the cell. So you can see that this is plasma membrane and it is pulled away from the cell wall. Okay, so the cell is in plasmolized. Alright, so the plant cell will also wilt. So normal shape of plant cell is turgid. So it always need hypotonic solution. 3.5.2 Facilitated diffusion. Facilitated diffusion, similarly to simple diffusion, it allows substance to move following its concentration gradient or down concentration gradient, and it do not need any ATP. However, compared to simple diffusion, facilitated diffusion needs transport protein. Remember, we have two types of transport protein, channel protein that is always open and carrier protein that have binding site and it change shape as the substance bind to the binding site. So channel protein provide the specific passageway for specific molecule to pass through. And it allows water or small ion to flow very quickly. So even though water can pass through plasma membrane by simple diffusion, but with the help of facilitated diffusion, more water can be transported across plasma membrane. So example of channel protein is aquaporin that is specific for water, and we also have ion channel. So we can see here, this is the movement of water. So water can still move across plasma membrane by simple diffusion. However, more water molecule can be transported into the cell by aquaporin. In ion channel, we have two different ion channel. Remember, channel do not change shape. So two different ion channel is lead ion channel. Another one is gated ion channel. So both channel do not change shape. However, in gated channel, it only have gate to control movement of substance. In analogy, just like your house, you have gate to control movement of anything in or out of the house. However, even though your gate is open, the house still do not change shape, right? So the gate can open or close in response to any stimulus. And we can have three different stimulus: chemical, electrical, or mechanical. So this is the example of gated channel and leak channel. Okay, so we can see that this is a gated channel. Sorry, this is example of gated channel, and this is example of ligand gated channel. When specific substance bind to this receptor, the channel protein will open. And you can see here, this is a voltage gated channel that when there is changes in voltage, the gate will open and the specific substance, the specific ion, can pass through the channel. One example of disease associated with channel protein is cystic fibrosis. Cystic fibrosis is a genetic disease, and it is characterized by abnormally thick and sticky mucus uh, in respiratory airways or in digestive tract. In cystic fibrosis, Cl minus protein transport is defect due to mutation, and Cl minus is usually transported associated with Na plus. However, since NaCl is accumulating in the respiratory tract, it affects antibiotic that kills bacteria in the lung. Therefore, you have accumulation of bacteria in respiratory airways or in digestive tract. So the bacteria will trigger cell lining to produce excess or abnormal sticky mucus, and this mucus will clog in the airways or in the digestive tract. Now we move on to facilitated diffusion using carrier protein. Remember, compared to channel protein, carrier protein can change shape. This carrier protein transport small hydrophilic molecule. Example is glucose and amino acid. And this shape changes in response to the binding and release of the substance. However, no ATP is required. This is glucose transporter, and this is the binding site, and it is specifically binding to glucose. So glucose will bind to the binding site. As the glucose binds to the binding site, it triggers the protein to change shape. And then the protein changes its shapes. Look at here, it already changes its shape. And then it will release glucose into the cell. Cystinuria is example of disease associated with carrier protein. It is an inherited disease due to too much amino acid inside the urine. Normally in the kidney, look at here, this is your nephron, this is the paroxysmal tubule. So normally cystinuria normally the amino acid is absorbed in the paroxysmal tubule. However, due to the carrier protein that transport cysteine is missing or defect, cysteine accumulate inside the urine. Supposedly, cysteine inside here, paroxysmal tubule, will be reabsorbed into the bloodstream. However, due to the missing of the carrier protein, cysteine accumulate inside the urine and it will form crystals. And this crystal will block the kidney. Probably it can block the ureter as well as bladder. So you can see this is your bladder. This is the stone that block the bladder. Probably it will also block the ureter here, ureter, or it will block the kidney itself.